All right, thanks, Henry. Um, so great to have this opportunity to talk about the work in my lab. And we developed optical imaging techniques to address biomedical questions that have been difficult or impossible to approach with traditional methods. So as you can see, we currently have three directions in the lab. Uh, in the first direction, we target on the unknown etiology of tubal ectopic pregnancy. Since it's so hard and challenging to study this life-threatening reproductive disorder, especially on how it forms. So our goal is to establish a new intravital in vivo imaging platform that integrates 3D structural, functional, and dynamic imaging together with optogenetic control to really address this technical hurdle in the ectopic pregnancy research. So the second direction is on biomechanical imaging, where we developed non-contact optical elastography methods. It is well known that the tissue elasticity or stiffness is a change of those properties is well involved in numerous pathologies such as cancers and cardiovascular diseases. So by imaging and measuring the elastic properties of tissue, we can enable early detection, better diagnosis, and advanced monitoring of treatment. So interestingly, stiffness is scale dependent. For example, at an individual cell level, metastatic cancer cells are softer than normal cells. But on the macro level, tumors appear to be stiffer or harder, which can usually be sensed by palpation. So this really indicates the significance or importance of probing and understanding biomechanics at different scales. Currently, oh, I can't do that. So, okay, currently existing techniques largely rely on atomic force microscopy that measures subcellular level stiffness, and also ultrasound and magnetic resonance elastography for the organ level analysis. So this leaves really a big gap here for microscale tissue level assessment. We are using light to develop techniques that can exactly fill in this gap, providing mechanical imaging of cells as a group together under their native environment. So in the example here, I'm showing that to understand the biomechanics of myocardial infarction, our method can provide not only high resolution mapping of a stiffness in the tissue, but also direction resolved elasticity in the healthy myocardium as well as infarcted region. So we are currently exploring new contrast mechanisms and expanding new applications, and also pushing this technique further for higher resolution and faster imaging speed. So in the third direction, we are collaborating with Irina's lab at Baylor College of Medicine to study the biomechanics of early mammalian cardiogenesis for better understanding of congenital heart defects, which is the most common birth defects. So specifically, my lab works on quantitative imaging of the hemodynamics shown here in the beating heart. And most recently, we have established a new approach for the pumping analysis in this tubular heart, especially on how particular flow patterns are generated. So currently, we are developing methods for spatial temporal analysis of the flow-induced shear stress on the endocardium to better understand how this, basically better understand this mechanical factor in the mechanical transduction involved in early heart development. So I think in the next 4 minutes or so, I'm going to into a little more details in the first direction. The mammalian oviduct, or can also be called as fallopian tube, is a tubular structure that connects the ovary and the uterus. There are primarily three reproductive events taking place in the oviduct: the transport of sperm and oocytes, fertilization, and embryo transfer. So as you figure out, there are a lot of transporting activities involved, so there need to be motors. There are two sources of force in the oviduct. One is the contraction of smooth muscle located on the wall. The other one is a bit of motile cilia on the epithelia of the lumen. So despite a long history of research, how these two motors or sources of force contribute or regulate the reproductive process inside the mammalian oviduct is still not clear. A primary reason is that most studies were based on in vitro or ex vivo tunnel settings, just like these histologies. As a result, today, very little is known about how a normal reproductive process looks like inside the mammalian oviduct. And in terms of disordered conditions such as ectopic pregnancy, it has been nearly impossible to investigate the dynamic and functional parts of this reproductive process that has been disrupted in the mammalian oviduct. So this is exactly the missing component of current reproductive research. And unfortunately, there has been no imaging tool allowing us to do that. So over the years, we have been continuously working to resolve this technical challenge and to make it possible to for in vivo studies of mammalian oviductal structure, function, and dynamics with a high resolution. And this includes, as shown here, microscale mapping of the Cilia bit frequency on the luminal epithelium, and in vivo 3D assessment of the oviductal smooth muscle contraction, and also the contraction wave propagation, as well as in vivo 3D tracking of sperm inside the oviduct ampulla, showing a number of never before seen sperm behaviors in the native fertilization environment. So most recently, we have achieved this first in vivo 3D imaging of the dynamics of oocytes and preimplantation embryos as they are transported through the oviduct. And in the example shown here, it shows that the cumulus oocyte complex has an unexpected circular movement inside the oviduct ampulla, and their trajectories are spatially distinct. So this 3D imaging approach really opens the door for in vivo studies of embryo retentions in the oviduct as a prerequisite for tubal ectopic pregnancy. And we're currently developing manipulation and interventional approaches that can be combined together with our imaging techniques to elucidate the functional cause of embryo retention in the mammalian oviduct. So that's all, and thanks for listening. Happy to answer any questions.
Well, thanks much, Sean. Very exciting work. I don't see any questions submitted. Let me try and ask perhaps a very naive question anyway. So when it comes to optical imaging, right, because of limited tissue penetration depth, unless you do endoscopy-based imaging, you can hardly produce the images that you are showing. So is that the technique that you used?
Well, yes, it's very right. So for optics, getting into deep inside the tissue is pretty hard. So first of all, we are working with the mouse model. So to get the knowledge out, basically with all the tools that already exist with mouse model. And also still to really approach to the reproductive organs in the mouse is still very, very hard. So what we did is we employed an intravital window that sort of can provide us the basically the light, provide us opportunity to deliver the light into and get the image out. So that's the basically the way we go inside the body. And as you said, there's endoscopic approaches that combine with optical imaging that can really give images inside the body.